Universe College of Nursing, Post-RN Biochemistry. Today we are going to discuss lipids. In the previous section, we have discussed major biomolecules which were carbohydrates and proteins. So the topic of today is called lipids, or we can say fats. Lipids from the Greek lipos, which means fats. These are heterogeneous class of naturally occurring organic substances which are insoluble in water and highly soluble in organic solvents like ether, chloroform, benzene and acetone. So the first point is that we have lipids which are non-polar. These are not polar molecules, that's why they are not soluble in water. They are only soluble in non-polar solvents, which are organic solvents actually, that are ether and chloroform, benzene, acetone. Chemically, lipids are various types of esters of different alcohols. So that means that basically lipids are made from the alcohols. One of them is alcohol, which is the major component. In addition to alcohol, there are some fatty acids. Some of the lipids may contain phosphoric acid, nitrogenous base and carbohydrates, which could be carbohydrates as sugars. So we have mainly lipids that are made up of two major molecules. Number one is alcohol and second is acid, that is fatty acid. So when acid reacts with alcohol, the product which is formed is called ester. So lipids are basically esters that are made up of alcohols and acids. So we have different types of lipids. Talking about the classification, mainly some of the lipids are called simple lipids, then other are called complex lipids, and the third class is called precursor, or we call it derived lipids. So the simple lipids are fats and waxes, while the complex lipids are phospholipids and glycolipids, while the precursor or the derived lipids, they are known as acyl glycerols, cholesterol or cholesterol esters. Fats, which is one of the simple lipids, these are made up of fatty acids that react with glycerol. Oils are fats in the liquid state. The oil that we use in our daily life, that oil is a fat which is a simple lipid. The next thing is waxes. These are also fatty acids with high molecular weight alcohols. So when a high molecular weight alcohol react with glycerol and fatty acids, that is known as waxes. The next class is called complex lipids. Complex lipids contain phospholipids. Lipids containing, in addition to fats and alcohol, another phosphoric acid residue. So the lipids which contain phosphoric acid, that are phospholipids. They have nitrogen containing bases and some other substituents, like we call them glycerophospholipids. The alcohol is glycerol, and then there are sphingospholipids in which the alcohol is sphingosine. And then we have glycolipids like glycosphingolipids. These lipids contain a fatty acid, sphingosine and carbohydrate. As you see, glyco means carbohydrates. Then we have sphingo, that is alcohol, sphingosine, and then lipid contains also acid, that is a fatty acid. The third thing is some derived lipids. So derived lipids, as already we talk about, that these are acyl glycerols, cholesterol, cholesterol esters, and some other complex lipids are lipoproteins. When a lipid combine with protein, these are called lipoproteins. Then we talk about that lipids are made up of two things. The first is the acid and the second is the alcohol. So the acid which we use in lipids is called fatty acid. Now fatty acid, these are big molecules. They may be saturated fatty acids or unsaturated fatty acids. Saturated have one single bonds, while the unsaturated fatty acids have double bonds or triple bonds. They are long carbon chains from 4 carbons to 36 carbons in one molecule. So when they are saturated, that means that all the carbons have single bond, and when they are unsaturated, that means they contains double bond between the carbon atoms. Now there could be mono unsaturated carbon atoms, that means there is one double bond, or there could be more than one double bond, that would be polyunsaturated fatty acid. So when there is one double bond, their general formula is CnH2n minus 1 and carboxylic acid. Oleic acid is the example. While there are polyunsaturated, that means they have more than one double bond, that are linoleic acid, linolenic acid, or arachidonic acid. So this is the example of an saturated fatty acid, where you can see that every carbon has one single bond between the carbon atoms. If there could be a double bond, that would be called unsaturated, but without double bond, they are saturated fatty acids. Now as you can see, there is a double bond between the two carbons, so that is oleic acid, that has one double bond, so we call it mono unsaturated fatty acid. And then there is another phenomena that take place, is called that isomerism. Isomerism is that when there is a same chemical formula but there is a difference in the structure, that is called isomerism, and there are two types, that is cis isomerism and trans isomerism. We would see the examples, that there are two carbon atoms with unsaturation, that means a double bond. So cis isomer is when this hydrogen atom and this hydrogen atom is on the same side, and this R group and R group on the same side. In trans, the hydrogen atoms, as you can see, they are on the opposite side, and the R group is also on the opposite side. So when the same atoms are on the opposite side, these are trans isomer, but when the same atoms are on the same side, these are cis isomers. As you can see in oleic acid, where you can see the cis transformation. Well, allylic acid, you can see the trans factors. So you can also see here that cis, these hydrogen atoms are on the same side, while in trans you can see these hydrogen atoms are on the opposite side. So there are positional isomers, that is a variation in the location of double bond along the unsaturated fatty acid chain produces isomer of that compound. So you can see that there is a double bond at this position and double bond at this position. These two molecules are exactly same to each other. The exception is that here the position of double bond is changed and here the position of double bond is changed, although both of the molecules are exactly same. This is called positional isomerism. Then let's discuss some of the physical properties of fatty acids. They have melting points of saturated fatty acid, and the melting point increases with increasing the number of carbon atoms. So when the fatty acid is of large size, the melting point is high. When the fatty acid is of small size, the melting point is low. Next is the solubility. Now the solubility of fatty acid increases with the double bond. So when there is increasing the number of double bonds, the solubility increases, and the solubility decreases due to the increase in methylene groups. Now formation of salts. Fatty acids, when they combine with salts like sodium, potassium, calcium and magnesium, they form soap. That means the soap that we use in our homes to wash our hands and face, this soap is actually made up of salt combining with the fatty acid, so they form soaps. And the detergents that we use to wash different clothes or we use to wash ourselves, these detergents are produced by the reduction of carboxyl group of the fatty acid, that produces alkyl alcohols that can be sulfated to form alkyl sulfates, and when the alkyl sulfates formed, these alkyl sulfates act as the detergents. Then coming to the ester, whenever the fatty acid reacts with the glycerol or any acid reacts with alcohol, ester is formed. Specifically in this case, 3 fatty acid reacts with a glycerol to form ester, that is triglyceride, with the emission of 3 water molecules. Then another property of the fatty acid is the formation of icosanoids. Icosanoid means some compounds with 20 carbon atoms. These compounds with 20 carbon atoms are called icosanoids. Now, prostaglandins are one of the examples. Arachidonic acid are also derived from the icosanoids, and thromboxanes and leukotrienes that play a very major role in the inflammation. These are formed from the icosanoids, which are actually fats. Now, these icosanoids are paracrine or local hormones. That means they act on their neighboring cells. Now, the prostaglandin is a 5 carbon ring originating from the chain of arachidonic acid. Another property is called hydrogenation. That means if there is an unsaturated or double bond fat, so there is a double bond fat, that means it has the potential to react. So whenever you add hydrogen into the unsaturated fat, that is called hydrogenation, and that is the way you convert oils to solid ghee, solid fats, the ghee that we use in our kitchen. So that is the called hydrogenation. The next is halogenation, that whenever you want to add a halogen atom, fluorine, chlorine, bromine, iodine, so that is called as halogenation, that whenever you add a halogen atom to an unsaturated fatty acid, that is called halogenation. And this property is used as the index of degree of unsaturation. That means how much a compound is unsaturated, that determines the degree of unsaturation. The next is the iodine number. That means if you have 100 grams of fat and you add iodine to that 100-gram fat, so the number of grams of iodine required to react with the double bonds in the 100 gram of fat, that number is called iodine number. Then oxidation. You know, the double bond in unsaturated fatty acids, these are oxidizable. That means they can undergo oxidation. Now the spontaneous oxidation, the oxidation which takes place by its own self, that is the spontaneous oxidation. That is a transparent coating on the surface of oil that is applied. This is called drying of oil. The oil gets dried. Specifically, that technique is used in manufacturing of paints or varnishes, that is done on the furniture. The next property is called rancidity. So when there are foods that are lipid rich and they are exposed to oxygen in the air, they may become spoiled or they become rancid. This is called rancidity. The unpleasant taste and smell that is associated with rancidity, whenever the food gets rancid or the food becomes unable to eat, the food that becomes unsuitable or unfit to eat, that becomes rancid. They result from the oxidative cleavage of the double bonds in the unsaturated fatty acids. That means the unsaturated fatty acid present in the food, there becomes oxidative cleavage of that fatty acid, which produces aldehydes or carboxylic acids of shorter chain length. They are highly volatile, so they have very unpleasant taste and very bad smell, and these compounds pass readily to air and then you can smell through your nose. To improve the shelf life of vegetable oils, shelf life means that whenever you put the vegetable oil on the open a bottle of vegetable oil and that becomes the shelf life of that vegetable oil. So that is known as shelf life, that whenever you are using oils in your kitchen for different purposes, for different cooking, so that is called as the shelf life, how much that oil could be suitable for how much time. Now to increase their stability at high temperatures, commercial vegetable oils are prepared by partial hydrogenation. That means there is a hydrogenation that is done to these oils, so their shelf life become increased and you can store these oils for longer duration of time. Now what actually this process is doing, it converts the cis double bonds in the fatty acid to single bonds. As hydrogenation is done, so they are converted to single bonds, the melting point of these oils is increased and they are more nearly solid at room temperature, they become ghee. So that is how their shelf life is increased. Then partial hydrogenation has another undesirable effect which is not desirable, that is not good, that some cis double bonds are converted to trans double bonds. That is not a wanted property. Now the dietary trans fats, that means that we use in our diet, they raise the level of triglycerols and of LDL. Now remember, there are two types of lipids. Number one is good lipid, that is HDL, high density lipid, and then there is LDL, low density lipid, which is bad. In our food, what we require is, we require increased HDL and less amount of LDL. That is what we require. But if the LDL is increased and HDL is decreased, then it can cause health problems like coronary heart disease. So that is unwanted effect. We need increased HDL and decreased LDL for good health. And then we have some essential fatty acids. So there are three polyunsaturated fatty acids called linoleic acid, linolenic acid and arachidonic acid. These three fatty acids are the essential fatty acids. That means they are must required in our body. We cannot synthesize these kind of fats. We have to take these fats from the diet. So in our diet we need these kind of fats. If these fats are decreased, or if they are deficient in the body, then there could be growth retardation and there could be deficiency syndromes which would be very devastating for the health. The next thing is docosahexaenoic acid, or we can call it DHA. Now what is docosahexaenoic acid? It is also a polyunsaturated fatty acid which is synthesized from alpha linolenic obtained from dietary fish oil, or it is also obtained from placenta, or it can be become from the milk, from the mother's milk. This fatty acid, it is present in high concentration in the retina, cerebral cortex, testis and sperms. It is very important for the development of brain and retina. That is called DHA, docosahexaenoic acid. Then there is alcohols contained in the lipid molecules. These alcohols are glycerol, cholesterol and higher alcohols like cetyl alcohol, or usually found in waxes. Now whenever there is OL in the end of the word, that is alcohol like glycerol, or it is also known as glycerine. Very simplest alcohol, trihydric alcohol, that means it has 3 hydrogen and it contains 3 hydroxyl groups in the molecule. Steroids and sterols. Now steroids are synthesized from the 5 carbon isoprene unit, part of a diverse collection of lipids called isoprenoids or terpenes. Now this ring, these 4 rings, these are steroidal molecules that are also lipid soluble, or you can say fat soluble. Now cholesterol. Cholesterol is also containing the 4 rings just like steroids. It is an important sterol that is present in the human body. We call it cyclopentanoperhydrophenanthine nucleus. That is the chemical name of the cholesterol. It has an OH group at the 3rd carbon. The name cholesterol is derived from the Greek word which means solid bile. Its melting point is very high, that is from 147 to 150 degrees centigrades. Cholesterol is very important for our human beings. It is a part of the cell membrane. It is a precursor of bile salts, male and female sex hormones. It is a part of vitamin D. It is linked to atherosclerosis, which is not good for the health. Bile salts, that is important in the lipid digestion. And this is, as you can see, a steroidal nucleus, that is steroid. You can see these 4 rings that are steroids. Also this is testosterone, that is the male sex hormone, which is a steroidal hormone. Progesterone, which is the female sex hormone, also a steroidal nucleus. And then 7-dehydrocholesterol, which is the precursor of vitamin D. So important sterol present in the skin. It differs from the cholesterol in having a double bond between the carbons 8 and 7 and 8. In man, the 7-dehydrocholesterol is obtained from the synthesis of cholesterol in the skin or in the intestinal wall. So when we are exposed to the skin or UV rays, sunshine, they change the 7-dehydrocholesterol to cholecalciferol, which is the vitamin D. Then we have ergosterol, that is not present in the human beings, that is present in the fungus, yeast. It is almost similar to the cholesterol, but that is a different kind of steroid, which is ergosterol, which is present in the fungi, not present in the human beings. Similarly, as you can see, bird feathers or pigments in the plants, with that are the compounds with long conjugating systems. They have also absorbed sunlight. Now the birds, they have pigments, and because of these pigments, birds have very beautiful colors of their feathers, that are like yellow or red by eating plant materials. These plant materials actually contain pigments like carotenoid or canthaxanthin or zeaxanthin, and the difference in pigmentation between the male and female birds, intestinal uptake and process of carotenoids. You can see those birds, because they eat these kind of plants and then they get pigments via eating the plants. Then we have lipoproteins. Lipoproteins are biochemical assembly that contains proteins and lipids. As you can see, lipo means lipid. A protein is a protein, that is a conjugated product of a lipid and a protein. And the lipids or derivatives may be covalently or non-covalently bonding, and that there could be a bond between the lipid and the protein. Many enzymes, transporters or structural proteins, antigens or adhesins, toxins, they are all lipoproteins actually, and they are very high density and low density lipoproteins of the blood. And there are transmembrane proteins of the mitochondria, chloroplast, and then bacterial lipoproteins. Now the lipoproteins constitute the petroleum industry also. They are very important in the petroleum as well. Now, like there are great oil tankers that travel through the oceans in the world, there are some chylomicrons in our body. Chylomicrons are large lipid rich transport vessels. What do? Ferry the dietary triglycerides in the main oil in the body throughout the circulatory system. That means throughout the blood they transport the lipids, and then they finally take the lipids to the liver and the chylomicron remnants. The very low density lipoproteins, that is not good for the health, VLDL, they are also like tankers. They also carry triglyceride in the liver to cells for energy and storage of fat. Now the LDL, which I told you not very good for the health, they are empty tankers. They only deliver cholesterol to the peripheral cells and liver. They have been offloaded. While the HDL, that is a very good fat for the health, what do? They clean up, crew, gathering up the excess cholesterol for transport back to the liver. Now, the cholesterol is used for the body for such functions like triglyceride transport by lipoproteins, maintaining the normal structure, integrity of the cell membranes, precursor for steroidal hormones, so they are used in the synthesis of hormones. But when they get in excess, they can also cause cardiovascular disease. Then there are some biological functions. We can get very high amount of energy from the lipids. So first of all, their energy storage, they can give you 9 kilocalories of energy. Phosphoglycerides, sphingoglyceride, they are steroids, very important structural components. Steroidal hormones, intracellular messengers. Then lipid soluble vitamins are there. Dietary fats act as carrier of lipid soluble vitamins, provide shock absorption and insulation. That means they can be shock absorber, they can protect you from the dangers. So that is all about the lipids. So in the next chapter we will discuss about the metabolism of lipids. That's all. If you have any question, you can ask in the comment section.